giving a talk today in this Autocatalysis seminar. I'm going to talk about, as Nicola said, about enumeration of autocatalytic subsystem in large chemical reaction networks. And this is a joint work with Thomas, who's here in the seminar as well, Peter, and also Nicola. And to my person, I'm a PhD student at Leipzig University in the bio group of Professor Peter Stadler. And so to give you a small overview of what I'm going to talk about, so first of all, I'm going to set some preliminaries, some notation we're going to need, and then I'm going to talk about two representations of autocatalysis. So these are autocatalytic matrices, and then I'm going to translate the properties of the matrices we're interested in into a directed bipartite König graph representation, which is also sometimes known as the metabolite reaction graph, MR graph, or species reaction graph, or SR graph. And then I'm going to show you how our formalism or our algorithm can be extended to enumerate autocatalytic cores very fast, but I'm going to just give a small introduction into this. Yeah, so whenever somebody talks about chemical reaction networks, there are different notions or different ways of thinking about chemical reaction networks. So for us, this is a set of species and a set of reactions, as he also said. And for each of these reactions, we can write a stoichiometric equation in that way. And then we can arrange the stoichiometric coefficients in a stoichiometric matrix. And then we get one representation of the chemical reaction network. But we can also get another representation. As I said, this is the König's graph or the MR graph, which is a directed bipartite graph with two disjoint sets of vertices. So we have metabolite vertices depicted here as circles, and we have reaction vertices, which are depicted as squares. And then we have two distinct types of edges. We have educt edges, which are from the metabolites into the reaction nodes whenever this metabolite is an educt for this reaction. And we have product edges. So we get an edge from a reaction vertex into a metabolite vertex whenever this metabolite is a product of this particular reaction. And this is how they can be visualized as a graph. And so I'm not going to give like a deep introduction into what autocatalysis is. I'm going to start basically with the overview of what we want to aim or what I want to talk about in this talk. So what we're interested in are finding a subset of autocatalytic matrices. And autocatalytic matrices are submatrices of the stoichiometric matrix, but enumerating them all would be computation infeasible. And this is why we're looking for a specific subset of these matrices. And these are so-called, or we call them autocatalytic child selection matrices with irreducible Metzler part. Among them, you find also the autocatalytic cores. The problem is, of course, in a graph, the stoichiometry is not encoded, and that's why we cannot find a direct representation of these matrices directly in the directed bipartite König's graph. But there is an equivalent representation for child selection matrices with irreducible Metzler part, for which we can then check are they autocatalytic or not. And these are so-called these are the so-called fluffles. And fluffles, they have specific properties. Fluffles are strong blocks which have the same number of metabolite vertices and reaction vertices, and each of the metabolite vertices has out degree 1, while each of the reaction vertices has in degree 1. And then the target or the goal becomes how can we enumerate these fluffles efficiently in order to find all of these child selection matrices with irreducible Metzler part. And for this, we're going to take advantage of so-called elementary circuits, so closed directed paths in the directed bipartite König's graph, which are themselves fluffles. And then we superimpose these elementary circuits in a suitable way by taking advantage of an equivalence relationship, which is defined on the reactant to reaction edges. And in this way, by taking advantage of this equivalence relationship, we can lower dramatically the computational costs that we need in order basically to find all of these matrices. So I'm going to explain all of this in more detail now. So let us start, but I want to say something before. before I start with the definition of an autocatalytic matrix, which you maybe all know, what I'm not going to talk about, or what is not included in this work, are explicit catalytic reactions. So an explicit catalytic reaction is a reaction when you have a species that appears both as a reactant and a product on both sides of the reactions. So, for example, the problem then is that these two reactions, they are represented by the same stoichiometric matrix. And then the stoichiometric matrix does not uniquely identify you the network anymore. And we, in the first work, so we excluded this case, but there is an extension to our work which deals with autocatalytic cores in reaction networks with explicit catalysis, but we postpone basically the treatment of this case to another work. And what I'm also not going to talk about today is the relationship between autocatalytic cores and another definition of autocatalysis, which is also around in the literature for some time now, which are the so-called reflexively autocatalytic F-generated sets, the so-called RAFs, which were introduced by Wim Hordijk and Mike Steele in 2004. This is current ongoing work in our group, but I'm not going to talk about this also today. Okay, so let's start with the definition of an autocatalytic matrix without explicit catalysis, as it was introduced by Alex Brockhaus et al. In 2020. And since we now exclude the case of explicit catalysis, a submatrix of our stoichiometric matrix is autocatalytic if we have a positive and a negative entry for each of the reactions. So we have a reactant and we have a product for each of the reactions. And we can find a strictly positive vector of reaction rates, such that if we multiply it from the right-hand side to our matrix, we get a strictly positive net increase of all of the species. And we're not so familiar with the autocatalytic framework. Here's an example. So for example, if we have x2 goes to 2x1, and then x1 goes to x2, then we get this stoichiometric matrix. And with this vector, we obtain a strictly positive vector of net increase of all species involved in the system. And then the next question, which arises very quickly, is the question, is this the smallest one I can find? And this leads to the definition of an autocatalytic core. And an autocatalytic core is just an autocatalytic matrix that does not contain a proper autocatalytic submatrix. And what we've seen in the example before, that is an autocatalytic core, because whenever I restrict myself to these positive entries, which I would need to find a strictly positive reaction vector, then I would not have a negative entry anymore. So I would not have a reactant for each of the reactions. Okay. And in a setting where all of the reactions are considered reversible, in the paper in 2020, Alex and his collaborators, they gave a complete characterization or classification of all of the autocatalytic cores into 5 different types depicted here. However, in our case, we assume that there can also be reactions that are irreversible. So we can have reactions that are reversible, and we can have reactions that are irreversible because the activation energy is too high, actually. And this is also meaningful in some biochemical reaction networks or some metabolic networks, such that this reaction would probably never happen in the backwards reaction. And we substitute a reversible reaction then by 2 reactions, a forward and a backward one. And then we also found in our results that then there can be other types than these 5 cores. Okay. And autocatalytic cores, it has been shown that they have very nice properties. So they are represented by square, reducible, and invertible matrices. And what is for our work now very important for this talk is that they have a representation. So when you reorder the columns of this matrix A tilde, then you obtain a matrix which has a strictly negative diagonal and a non-negative off-diagonal entries. And a matrix that has non-negative off-diagonal entries is called a Metzler matrix. And when we have a look at our core from before, and now it's called S tilde, if we just switch these two columns, so we switch the two reactions and we reorder the columns, then we obtain a representation as a Metzler matrix. And this Metzler representation, especially in the case where we exclude explicit catalysis, that shows us another property which has been shown totally with a total different approach for autocatalytic cores, and this is that each of the reactions in an autocatalytic core has one reactant and one reactant only, and each of the metabolites involved is a reactant for exactly one reactant, for one reaction. And this, to formalize this in a concept, we can take advantage of so-called child selections. And child selection is a triple composed of a subset of the species, a subset of the reactions of identical size, so of size k, and we can find a bijection from the subset of the species into the subset of reactions such that the species chooses it's a pertinent reaction for which it is actually a reactant for, so that we have a stoichiometric EDA coefficients of the species and the image of the species under the bijection, which is strictly positive. And then we call this a child selection bijection. And maybe this is more easy to understand at a graph. So for example, in this case here, in this reaction network here, when we just look at this subnetwork, then A can only choose R1, B can only choose R2, C can only choose R3. And this is also actually a type 3 autocatalytic core. In autocatalytic cores, there will be always only one child selection which is possible, but there might be other ones in general. So in general, the child selection, the bijection might not be unique. Okay, and maybe to sum up this first part, so I'm talking about reaction networks without explicit catalysis and where we have reversible and irreversible reactions. And with the assumption that we exclude explicit catalysis, our autocatalytic matrices uniquely identify as autocatalytic subsystem in the network, in the graph. And we have also seen that autocatalytic cores, they satisfy basically a lot of the properties we were looking for. So the inclusion minimal autocatalytic matrices, we know that they're irreducible, and they have a representation as the Metzler matrix, which is also a child selection matrix. And at the beginning, we actually set out to find all irreducible autocatalytic Metzler child selection matrices. But as I pointed out at the beginning, it turned out what we actually found were all autocatalytic child selection matrices with irreducible Metzler part. And you may ask, okay, what is a Metzler part of a matrix now? So whenever you have a matrix which is here as a child selection, represented as a child selection matrix, and you find a negative off-diagonal entry marked here in red, then you just turn these entries into zeros and you obtain what we call the Metzler part. And if this Metzler part is irreducible, then we call this matrix a child selection matrix with irreducible Metzler part. And these negative off-diagonal entries, they become important later on, and I'm going to explain why. So let us now try to translate, after we set the goal of what we're looking for, translate all the properties of the matrices into our König's graph representation. And we start with the child selection. And we can look at this from two different perspectives. And we start by asking the question, so whenever we have a subgraph of our König's graph, can this be child selective? So does there exist a map from the subset of the species into the subset of the reaction vertices that I can actually find a bijection from the subset into the subset of the species into the subset of the reaction vertices, and then this triple becomes a child selection. And I've depicted here or shown here many examples of which actually only A and G are child selective because B, C, D, and E they somehow violate that the number of the species vertices have to be identical with the reaction vertices. And here in F, this is an example where X2 and X3 can only choose the same reaction. But in A, you can see, okay, we have elementary circuits. These are elementary circuits, which also become important later on. And disjoint unions or not connected unions of elementary circuits are child selective. And what you've seen here in G is what you've actually seen already before. This is a type 3 autocatalytic core. Of course, this one should be child selective. And there is a nice characterization in graph theoretical terms of child selectiveness. So a subgraph is child selective if and only if the subset of the reactant to reaction edges contains a perfect matching. So whenever I can basically uniquely identify metabolite vertices with reaction vertices. And of course, there is a one-to-one correspondence between perfect matchings and child selections. And here you can basically see two of them. The green one is one, but there is also another one, which is the blue one. And then, as I said, we can also have a look from the other side. So given a child selection, we can look at two different kind of induced subgraphs. So we can have a look at the vertex-induced subgraph induced by the vertices of the species and the reactions, which are for this child selection that we obtain this vertex-induced subgraph. And here you can see that the off-diagonal, so this negative off-diagonal entry marked in red here, that yields an additional species to reaction edge. But we can also look at the child selection-induced subgraph, as we call it, which has only those reactant to reaction edges that are defined by the child selection or by the corresponding perfect matching. And then we obtain the subgraph here. And then we basically lose this additional reactant to reaction edge. And then you can see basically then that this graph here is represented by the Metzler part of the child selection matrix here. And we may also observe at this point that each of the species vertices has out degree 1, and each of the reaction vertices has in degree 1, which is basically just what we wanted to have, and what is induced by that we just looking at the child selection induced reactant to reaction edges. And interestingly, looking back again from the other side, we obtain that the converse is also true. So if you have a subgraph that has the same amount of species vertices and reaction vertices, and every species vertex has out degree 1 and every reaction node has in degree 1, then this K prime, the subgraph, is actually child selective with a child selection, what we would expect it to be. Okay, so we translated child selection into a graph setting, and now the question becomes, okay, what is about this irreducibility? And in graph theory, whenever you have a digraph, then this digraph is strongly connected if and only if its adjacency matrix is irreducible. So reducibility is somehow connected to, is connected to or associated to strongly connectedness of a graph, of a digraph. And since we have these square child selection matrices, we ask the question, okay, is there some kind of notion also, is this also somehow true for our child selection matrices? And again, now the Metzler part comes into play. So again, let us look at this child selection matrix here, which is represented by this graph. And I also marked you this additional edge, which is induced by this negative off-diagonal entry. And to get from x1 to x3, there are basically two ways of going via reactions. And the one is by R1 to x3, and the other one is going via R2 to x3. For the other vertices, you can only go via one reaction. And then the Metzler, if I remove this edge, which is then encoded by the Metzler part, then I have only one opportunity of going from x1 to x3. And then this matrix is nothing more if you remove the diagonal entries than the adjacency matrix of the associated metabolite graph. So when I remove all the reaction vertices and I just have a graph which is defined on the metabolite vertices such that I can go from a metabolite to another metabolite whenever this first metabolite is a reactant and the second metabolite is a product for the same reaction. And since there's only one way of going from metabolites to other metabolites, this is the idea also for the proof. We not only get that for a child selection matrix, the child selection induced subgraph is a strongly connected graph if and only if it's the Metzler part of its child selection matrix is irreducible. No, it's also a strong block. That means that the underlying undirected graph is too connected. And to now summarize these results in a proposition, we obtain what we call a fluffle, or what we call fluffles. So if we have a subgraph, and then there is a child selection with child selection matrix that has an irreducible Metzler part, such that our subgraph is actually the child selection induced subgraph if and only if we satisfy what we now worked out, what we obtained as results. So we have the identical number of species and reaction vertices. Each of the species vertices is out degree 1, each of the reaction vertices is in degree 1, and G is a strongly connected block. So, and as I said at the beginning, elementary circuits are fluffles. So we call these subgraphs fluffles that satisfy 1, 2, and 3. And also, of course, the autocatalytic core type 3, we've already seen a couple of times, is a fluffle. What is not a fluffle is whenever I violate some of these conditions. So for example, here I have 3 reaction vertices and 2 metabolite vertices, then I do not get a fluffle. And then there's a nice result in the literature about strong blocks. And a graph is a strong block if and only if it has a directed open ear decomposition. And what is an open ear decomposition? So maybe we do it here at this fluffle. An open ear decomposition is basically a path from one vertex to another one, where each of the vertices on that path, they just has these vertices, they have in degree 1 and out degree 1. And there's also another path from the terminate vertex to the starting vertex. And for the fluffles, we get a specific open ear decomposition, directed open ear decomposition, which starts always in a metabolite, in a reaction vertex and ends in a metabolite vertex. And so the question was, if this characterization holds, can we also revert it? So can we do the inverse? Because we know that elementary circuits are fluffles. Can we start with elementary circuits and then stick elementary circuits together, so superimpose elementary circuits in order to obtain fluffles? And this is true, but we have to make sure, so if we start with a fluffle and add an elementary circuit, that the union is only a fluffle, so if and only if each connected component of the intersection of both of these graphs is a path that starts in a metabolite and ends in a reaction vertex. So what we have to obtain is that we have to get this inner part. And so taking these two elementary circuits, they share the A to R1 path. So it's a metabolite to reaction path, and sticking them together is what we obtained, what we've seen earlier, that we get a fluffle. And this can get arbitrarily complicated, of course. So you can add another elementary circuit down here, which shares the C to R3 path. Or, as I said, each of the connected components of the intersections has to be a path that starts in a metabolite that ends in a reaction vertex. So you can also have disconnected, so not connected components of the intersection here, A to R1, and then the green one is taking this deviation, the blue one this deviation, and then D to R4 is again the common path. And then you get, again, here a fluffle. Okay, So then the question, if we can compose, or if we can create fluffles by suitable superimposition of elementary circuits, the question is, can we find them all in such a way? And yes, this is true. So for each fluffle, there exists a set of elementary circuits, which we call circuit nets, such that the union of these elementary circuits returns us the fluffle. The problem is that many circuit nets generate the same fluffle. So for example, here C1 and C2, where C1 is composed of the green, the blue, and red elementary circuit, and C2, which is composed of this Bordeaux and this teal elementary circuit, C4 and C5, both gave us the same fluffle. So we came first up with the idea, okay, of introducing a fluffle equivalence that says that two circuit nets are basically equivalent or the same if they share the same, if the union of the elementary circuits within these circuit nets share the same edges. However, we did not set out actually to enumerate fluffles. We did set out to enumerate actually child selection matrices, or autocatalytic child selection matrices. And we can observe whenever we have all the E1 edges, so all the educt edges in our matrix, the rest is basically induced. The rest of the graph is induced, all the product edges. And this is why we came up with the notion of a child selection equivalence relationship, which is defined only, not on all of the edges, but only on the educt edges. So two circuit nets, they are basically considered equivalent if their union, if the union have the same educt edges. And strikingly for this is a type 5 autocatalytic core, by the way, 28 out of 31 circuit nets belong to the same equivalence class. Okay, so this somehow suggests that we can reduce computational costs. So the last thing we had to ask were, okay, if we now have representatives of one equivalence class and a representative of an elementary circuit child selection equivalence class, if we stick them together, do we get basically the same as if we would stick together two other representatives of both classes? And this is actually the case. So for example, here, this example has to be modified because actually C4 is already giving us the whole fluffle, but C4 combined with C3 is the same as C5 with C3. But actually this does not make such a difference for elementary circuits, as you can see here on the right-hand side. So the number of elementary circuits, number of equivalence classes, this is for an E. coli core model. They're basically identical whenever you have to have some kind of type 5 core, some generalization of a type 5 core, because there you have two elementary circuits which are spanning the whole fluffle or the whole core. But it makes a dramatic difference if you look at larger autocatalytic subsystems, which you can see here on the right-hand side. So the number of equivalence classes that had to be enumerated in order to find them all were dramatically smaller than enumerating all the fluffle equivalence classes. Yeah, and with this, we set out basically to apply this also to metabolic networks. And these results I do not want to show here because of also for the time. But what I want to point out is a little bit that there is some kind of problem with this approach, because enumerating elementary circuits can get very expensive. And if you're interested only into enumerating finding autocatalytic cores, then you see that the difference between the elementary circuits you have to enumerate and the cores you end up with are for different kind of species, Helicobacter pylori, Staphylococcus aureus, or other ones, can get dramatically large. So the question was, can we do something about this? And this is what we did in the follow-up work. Yes, we can. And what we did is that we basically observed, okay, each of the cores is represented by a Metzler matrix, so there's no negative off-diagonal entry. And this translates into, okay, we do just want to have for each metabolite, we just want to have one metabolite to reaction edge, and also for the reaction edge, for the reaction vertices. So we do not want to have elementary circuits, because whenever we have an elementary circuit that encodes a matrix, a child selection matrix with a negative off-diagonal entry, we would never get rid of this negative off-diagonal entry. And this is why we came up with the idea of enumerating instead MR chordless elementary circuits. So elementary circuits that do not have any shortcuts from metabolites to reactions in the circuit. And all the metabolite to reaction edges are located along the circuit. And this dramatically, again, reduces the amount of circuits we have to enumerate. And then we can see actually for Helicobacter pylori, Methanosarcina, barkeri, and also Staph aureus, we've been able actually to enumerate all of the autocatalytic cores. Okay, and with this, and also with respect to the time, I want to sum up. So we found a representation of child selection matrices with irreducible Metzler part as fluffles, as I've shown you. And we can also enumerate them in a somewhat efficient way via this notion of child selection equivalence class. And for everybody who's interested in using or trying it out, you can find a tool which is implemented in Python on my GitHub page, which is called Autocatalytic. And there's also the smaller version I've shown you at the end with the MR chordless circuits, Autocatalytic, which is currently but under this name because for the publication, for the revision of the publication, we left it here before we're going to merge it. And as I said at the beginning, we also have now a framework including explicit catalysis and translating now Autocatalytic, which is implemented in Python, into C++, because in Python, whenever you want to have real parallelization, you need to copy large data structures such that the different processes can access them separately. And we also, as I said, trying to compare now the two definitions of autocatalysis. So we're comparing the definition of autocatalysis I've shown here to the RAF framework. And we also have current ongoing work with collaboration partners, which have chemical reaction networks, for example, on carbon fixation. Yes, and with this, I want to thank Thomas, Nicola, and Peter, and also the German Network for Bioinformatics Infrastructure for providing us with additional computational resources, the SEC AI Consortium for travel funding, and also the Atomic and Level Notice Fund for additional funding. And of course, I thank you all for your attention. Hopefully I can answer all the questions you might have.